Statins, from fungal metabolites to modern cholesterol-lowering drugs, discovery, medicinal chemistry, synthesis, and mechanism of action. The discovery of statins represents one of the greatest achievements in modern medicinal chemistry and cardiovascular pharmacology. Since their introduction into clinical practice during the late 1980s, statins have transformed the prevention and treatment of atherosclerotic cardiovascular disease, ASCVD, substantially reducing cardiovascular morbidity and mortality worldwide. Today, statins remain the first-line pharmacological therapy for hypercholesterolemia, with hundreds of millions of prescriptions written annually. Unlike many synthetic drugs developed through rational molecular design, statins originated from the systematic screening of microbial natural products. Their discovery highlighted the enormous therapeutic potential of fungal secondary metabolites and demonstrated how natural products can serve as templates for highly effective medicines. The pioneering work of Japanese biochemist Akira Endo initiated an entirely new class of lipid-lowering agents by identifying naturally occurring inhibitors of 3-hydroxy-3-methylglutaryl. Coenzyme A, HMG-CoA reductase, the rate-limiting enzyme of cholesterol biosynthesis. Subsequent advances in medicinal chemistry, fermentation technology, semi-synthetic modification, and total synthesis produced successive generations of statins possessing greater potency, improved pharmacokinetics, and superior clinical efficacy. The search for cholesterol biosynthesis inhibitors. During the 1960s, treatment options for hypercholesterolemia were extremely limited. Therapeutic agents such as nicotinic acid, fibrates, and bile acid sequestrants modestly lowered plasma cholesterol, but often led to poor patient compliance due to gastrointestinal side effects and limited efficacy. Meanwhile, epidemiological studies, including the Framingham Heart Study, have firmly established elevated low-density lipoprotein cholesterol, LDLC, as a principal risk factor for coronary artery disease. A breakthrough occurred in 1966 when HMG-CoA reductase was identified as the rate-limiting enzyme in the mevalonate pathway responsible for cholesterol biosynthesis. This discovery immediately suggested that selective inhibition of this enzyme could provide an effective strategy for lowering endogenous cholesterol production. Akira Endo, then a researcher at Sankyo Company Limited in Tokyo, recognized the remarkable analogy between cholesterol biosynthesis and microbial competition. Inspired by Alexander Fleming's discovery of penicillin, Endo hypothesized that fungi might naturally produce metabolites capable of inhibiting sterile biosynthesis in competing microorganisms. Such compounds would likely target HMG-CoA reductase, because many microorganisms require mevalonate-derived sterols and isoprenoids for growth. This hypothesis was remarkably visionary because, at that time, no naturally occurring inhibitor of cholesterol synthesis had been identified. Akira Endo's discovery of compactin. Beginning in 1971, Endo and colleagues initiated an extensive screening program involving more than 6,000 fungal strains isolated from soil, food products, and decaying vegetation. The screening assay measured inhibition of cholesterol synthesis using rat liver microsomes, incorporating radiolabeled acetate into cholesterol. Among thousands of fungal cultures, a strain of Penicillium citrinum isolated from rice exhibited exceptional inhibitory activity. In July 1973, Endo's group isolated 3 active metabolites, with the most potent designated ML236B, later renamed compactin or mevastatin. Structural analysis revealed an extraordinary resemblance between compactin and the tetrahedral intermediate formed during HMG-CoA reduction. This molecular mimicry immediately suggested competitive inhibition of HMG-CoA reductase, establishing the first direct inhibitor of cholesterol biosynthesis. The discovery represented one of the earliest examples of successful, target-based natural product screening in pharmaceutical research. Early preclinical development. Initial animal studies produced mixed results. Rats demonstrated little reduction in serum cholesterol because rodents naturally maintain low LDL concentrations and rapidly compensate for reduced cholesterol synthesis. Recognizing these physiological limitations, Endo expanded studies to dogs, rabbits, monkeys, and chickens. These experiments consistently demonstrated significant reductions in plasma cholesterol, confirming that compactin effectively inhibited hepatic cholesterol synthesis across multiple species. An important milestone occurred in 1978 when physician Akira Yamamoto administered compactin to a young patient suffering from severe familial hypercholesterolemia. Within only 2 weeks, total cholesterol concentrations fell dramatically from approximately 1,000 milligrams per deciliter to 700 milligrams per deciliter, providing the first clinical proof that inhibition of HMG-CoA reductase could successfully lower cholesterol in humans. Despite these encouraging findings, development of compactin temporarily stalled following toxicology studies reporting lymphomas in dogs receiving extraordinarily high doses for prolonged periods. Later analyses demonstrated that these doses exceeded therapeutic human exposures by nearly 200-fold, suggesting the findings were unlikely to reflect clinically relevant toxicity. Nevertheless, these safety concerns delayed commercialization. Parallel discovery of lovastatin. While Sankyo investigated compactin, an independent research team led by Alfred Alberts at Merck isolated another potent HMG-CoA reductase inhibitor from Aspergillus terreus in 1978. Initially named mevinolin, the compound was later shown to be chemically identical to monocolin K, independently isolated by Endo from Monascus ruber. Following comprehensive toxicological evaluation and large multicenter clinical trials, mevinolin became lovastatin, the first statin approved by the U.S. Food and Drug Administration, FDA, in 1987. Lovastatin established proof that selective inhibition of cholesterol biosynthesis could substantially reduce LDL cholesterol while exhibiting an acceptable long-term safety profile. The landmark approval fundamentally transformed cardiovascular medicine. Evolution of statins through medicinal chemistry. The success of lovastatin stimulated intense medicinal chemistry efforts to improve potency, metabolic stability, tissue selectivity, and oral bioavailability. The first generation consisted of naturally occurring fungal metabolites: compactin, mevastatin, lovastatin. Semi-synthetic modification of these natural products yielded second-generation statins, including simvastatin, produced by introducing an additional methyl group into the side chain of lovastatin, thereby increasing lipophilicity and inhibitory potency. Pravastatin, generated by stereospecific microbial hydroxylation of compactin using Streptomyces carbophilus, became considerably more hydrophilic, reducing extrahepatic distribution and improving tolerability. Subsequent medicinal chemistry efforts replaced the rigid decalin nucleus characteristic of fungal statins with heteroaromatic scaffolds while preserving the essential 3,5-dihydroxyheptanoic acid pharmacophore responsible for HMG-CoA reductase inhibition. These fully synthetic superstatins include fluvastatin, indole-CoR, atorvastatin, pyrrole-CoR, cerevastatin, pyridine-CoR, rosuvastatin, pyrimidine-CoR, pitavastatin, quinoline-CoR. Among these agents, atorvastatin and rosuvastatin exhibit the greatest LDL-lowering efficacy owing to stronger enzyme binding and prolonged hepatic activity. Atorvastatin, despite exceptional potency, was withdrawn worldwide in 2001 because of an unacceptable incidence of severe rhabdomyolysis. Biosynthesis and industrial production. Natural statins are produced through fungal polyketide biosynthesis. In Aspergillus terreus, lovastatin biosynthesis is orchestrated by a gene cluster containing approximately 18 genes. Two multifunctional polyketide syntheses, LOV-B and LOV-F, assemble the non-acetide and diketide precursors from acetyl-CoA and malonyl-CoA. Additional tailoring enzymes, including LOV-A, LOV-C, and LOV-D, catalyze oxidative modification, reduction, and acyl transfer reactions that ultimately yield lovastatin. Industrial production relies primarily on fermentation using Aspergillus terreus. Solid-state fermentation generally produces substantially higher yields than submerged fermentation because of enhanced expression of key biosynthetic genes, particularly the transcription factor LOVE. Modern strain improvement employs mutagenesis, metabolic engineering, and optimization of carbon and nitrogen sources to maximize production efficiency. Semi-synthetic statins are subsequently manufactured through selective chemical or enzymatic modification of naturally produced intermediates. For example, simvastatin is synthesized by acylation of monocolin J, derived from lovastatin, whereas pravastatin is produced through stereoselective hydroxylation of compactin by microbial cytochrome P450 enzymes. Mechanism of action. All statins share a common pharmacophore, consisting of a 3,5-dihydroxyheptanoic acid moiety that closely resembles the tetrahedral intermediate formed during reduction of HMG-CoA to mevalonate. This structural similarity enables statins to act as competitive and reversible inhibitors of HMG-CoA reductase. Crystal structure analyses have demonstrated that the dihydroxy acid group forms multiple hydrogen bonds within the catalytic pocket, while hydrophobic interactions between the statin ring system and adjacent residues further stabilize enzyme binding. Rosuvastatin and atorvastatin possess additional polar and fluorophenyl interactions, explaining their exceptionally high affinity. Inhibition of HMG-CoA reductase decreases intracellular cholesterol synthesis, lowering hepatic cholesterol concentrations. Reduced intracellular cholesterol activates sterol regulatory element binding protein 2, SREBP2, which upregulates expression of hepatic LDL receptors. Increased receptor density accelerates clearance of circulating LDL particles, resulting in substantial reductions in plasma, LDL cholesterol, typically between 20 to 60%, depending on statin potency and dosage. Beyond lipid lowering, statins exhibit numerous pleiotropic effects, including improved endothelial nitric oxide production, stabilization of atherosclerotic plaques, attenuation of oxidative stress, suppression of vascular inflammation, inhibition of smooth muscle cell proliferation, and reduced thrombogenicity. These effects contribute significantly to their cardiovascular protective benefits. Clinical impact and future perspectives. Since the approval of lovastatin in 1987, statins have become among the most extensively studied and widely prescribed drugs in medicine. Landmark clinical trials, including the Scandinavian Simvastatin Survival Study, 4S, the West of Scotland Coronary Prevention Study, WASCOPS, the Heart Protection Study, HPS, and the Jupiter trial, established that statin therapy significantly reduces myocardial infarction, ischemic stroke, cardiovascular mortality, and all-cause mortality across both primary and secondary prevention populations. Current research continues to optimize statin therapy through pharmacogenomics, precision medicine, combination therapies with ezetimibe and PCSK9 inhibitors, and improved understanding of statin pleiotropy. Despite the emergence of newer lipid-lowering agents, statins remain the cornerstone of dyslipidemia management because of their unparalleled efficacy, safety, extensive clinical evidence, and cost-effectiveness.